I think we can start the seminar. So welcome everyone. I hope you've had a great day or are about to have a great day. So welcome to this week's Paleoprax seminar. This week we have a talk by Arindam Chakraborty from the University of Malaya in Malaysia. And his talk will be about past environment of the Emerald Islands of India, evidence from the white and black fossil sites. So just if you're new here, a quick discussion of the format of today's seminar. So first, we're having a little welcome and announcements, which was we were doing today, right now. Then we'll have the talk, which will take around 30 minutes. And after that, you will have some time to ask questions. So please think about your questions during the talk. And please don't forget to send your questions via the chat to the questions host at Paleoprax. He will then I will read them out in the chat. So today we likely won't have a tea time because Arindam has already been very nice that he wants to give this talk right now, because for him it's actually almost midnight. So it will depend a bit on the time. So a bit more housekeeping. So Paleoprax values the participation of all folks interested in the paleosciences. And please remember to abide by our code of conduct during today's seminar. So if you are in this link, you should have signed off our code of conduct. If you haven't, then please go to our website and still fill it out. Sorry. Please mute yourself for the duration of your talk. You should be muted already, but if you find yourself muted to unmute during the talk, then please mute yourself again to avoid any audio distractions. And then again, you can ask the questions by sending them to the questions Paleoprax host, or by raising your hand function on Zoom if you want to ask the question yourself. Also, if you have any technical issues, you can also send it to the questions at Paleoprax host. And we now have closed captions built into Zoom. You can use the CC button automatically to show or hide them. So do that if you want to. Then we're always looking for interesting people to give interesting talks here. So if you know someone who you think that would give a great talk, please nominate them via the form, which will also appear in the chat. You can also self-nominate if you want. Then we also would really appreciate it if you can fill out our weekly feedback form for mostly demographic information. It's all anonymous and the link will also be posted shortly in the chat. So a bit more about today's speaker, Arindam Chakraborty. Arindam did his master's in marine biology at the Pondicherry University in India. And then he followed up with a PhD in marine micropaleontology at the Birbal Shani Institute of Paleosciences in India. He's held several postdoc positions, first starting as a National Postdoc Fellow at BSIP in India, then as a Birbal Shani Research Associate also at BSIP in India, and finally as a postdoctoral researcher at the Institute of Earth Sciences and the Academia Sinica in Taiwan. And currently he is a senior lecturer at the Department of Geology at the University of Malaysia. And he's also the supervisor of the Geology Museum in the same department. So now without further ado, I would like to invite Arindam to share his screen and start his presentation.
Thank you, Nina, for introducing me. And I hope everybody can see my screen. Can everybody see my screen?
Yeah, we can see your screen.
Okay. So, first of all, thanks to Paleoprax team for inviting me for giving a talk in this Paleoprax seminar series. So today I will be mostly talking about the past environment from the Emerald Islands of India. So, and it's the evidence from the white and black box fossils. So from the title only, you can see that there are different terms which are which I will be describing you later on. So the first one is the Emerald Islands, and next is the white and black box fossils. So first we will see our study area, where you can see that this is the Indian Ocean and here is the Pacific Ocean. So this is India and this side is Myanmar and then Thailand, Malaysia, and this is the Indonesian part of Java and Sumatra. So the slides may be a bit slow, the internet. Yeah. So here you can see we are seeing the study area. So this one is India and Bay of Bengal here, and we are zooming in into the Andaman and Nicobar Islands. So here I will show you. So this region is tectonically very active actually. So this is a part of the volcanic island arc system, and it's been formed by the collision of the Indian and the Burmese plate. So this side is the Burmese plate and this side is the Indian plate. So it lie along the Sunda Arc Trench. So this is the Sunda Arc Trench completely, and this island is a result of the uplift of the oceanic crust and its formation of the volcanic arc actually. So this is a subduction region where the Indian plate goes down to the Burmese plate, and this started this subduction started in the late Mesozoic after the breaking of the Gondwana land generally. So the main ridge, this is the main ridge, the island ridge, which started rising during the end of Oligocene, and there is the unconformity after that, and then during the Cenozoic in the sediments of the Neogene sediments are then and lie over the four-layer group that I will talk to you about in detail later. So this side you can see the back arc basin here. So this is the volcanic arc where there is some active volcanoes still. The barren island here and Narcondam Islands, which are the barren volcanic islands, still it is active. And this one here is the four arc basin, and here is the four deep, and this is the trench actually, and this is the mainly the accretionally prism and the inner slope beside. So so this part till here it will be the Andaman Islands, and this down is the Nicobar Islands. So together it is known as Andaman and Nicobar Islandic group actually. So the next slide.
Arindam, I think
Yeah.
maybe if you turn off your video, the connection might be a bit better. The audio is just a little choppy.
Can you hear me clearly now?
Yeah, I think that's a bit better. Thanks.
Okay. So here you can see I talked to you about the Emerald Islands. Why I put that name actually. So this everybody knows the gemstone, which is known as emerald. It's green in color, and you can see the water here in these islands are really looks like emerald gemstone. So you can see these are different beach where people come as a tourist, and it's a turquoise blue water and completely empty beach where people can have a good holiday. So I named it as a Emerald Island. So in between the slides, it will be a little bit lagged, so please don't worry. Yeah, so I will be talking mostly about, because this island has been the deposition started from the late Cretaceous onwards, but I will be talking mostly today about mostly about Neogene, and where the major events on Cenozoic most of you all already know, but I just want to recap it. Like the KPG mass extinction event occurred here in the Cretaceous Paleocene boundary, and then the rise of the mammals and decline of the reptiles, and changes in from the climate from the greenhouse down here to the ice house climate throughout the Cenozoic, and the movement of the continents which to the present positions. And in the Neogene, there was complete disappearance of the Tethys Sea and the upliftment of the Himalayas, which formed. So, and from this, the sediments from these Himalayas are brought by the mighty rivers Ganges and the Brahmaputra, which forms the enormous Bengal deep sea fan. So, and the major changes of the oceanic circulation occurred, and permanent ice sheet formation in the Antarctic in the South Pole occurred. And continent generally attained the present position from that time actually. So this is the geological cross-section of the Andaman Islands. So here is the main island ridge, the main ridge here. And here is the western side of the island in the Bay of Bengal. And this right side is the eastern side of the island towards so this side is Indian coast and this side is the Thailand coast. So mostly this part is known as the Andaman Sea and this one is the Bay of Bengal. So you can see the number 1 is the Indian plate, which is going down, subduction zone, and number 3 is the Thai-Burmese plate, which is on top of the Indian plate. And we have the volcanic arc, which is above the seawater, and some of the sea mounts are associated with that underwater also. So mostly by different and workers, it has been mentioned that some mentioned that it has been opening. This island was opened up during the late Miocene, and some told that. But this is one of the recent study that in 2005, which concluded that the openings was of 2 times. One is in the late Oligocene to middle Miocene, and another was during the late Miocene to the early Pliocene. So here is the paleogeographic map. You can see during the Eocene time. 1 minute. So this is the Eocene time during the 50 million years when the Proto-Himalayan stage, the India was colliding with the Eurasian plate here, and the Tethys Sea was closing generally, and the formation of Himalaya started from here. And here you can see the Eo-Himalayan stage where the Himalayan belt started forming, and here is the Bengal fan, the blue in color also started, because when the Himalayan started, the rivers formed from here, and the terrestrial sediments are been brought by the rivers to the sea here. So the Bengal fan is formed, and during the Oligocene time, Miocene time in 15 MA, you can see more the Bengal fan is extended towards the down around to the 90s reach here, and this is the archipelago group, and this is the trench here in the red in color, and this one is the fan, that is the Irrawaddy fan. So this Bengal fan is generally from the rivers from the Indian side, the Ganges and the Brahmaputra, and this one, the green in color, is the from the Irrawaddy river from the Myanmar side. So this is the present day how the island looks like. So it is just here on the east coast of India. And these are the distributions. So I will be mainly talking about the red color sediments. That's the Neogene sedimentation. So you can see here, some are here, but it's very hard to get the sediments from this because most of the islands are under reserve forest. And by the way, I just want to introduce you. There is a very rich and diverse history of this island. They have around the indigenous tribes, like the Great Andamanis, the Onge, the Jarwa, and the Sentinelese. So who lived here for thousands of years and relying just on hunting, fishing, and gathering their livelihoods without and they are isolated from the so-called developed world actually. So they still now, some of the like the Jarwa, the Sentinelese, they don't be in contact with the local people, and they don't wear any clothes. It's like the original tribes that you see in the Amazon. So similarly, we also have in this island. So it's very hard to, because this is the reserve islands where the tribes stay. So we are not allowed to go inside. So the collecting sediments from this part is really hard. So we have some some of the sediments from this island, from Havelock, Neil, and some of here, and down from here is the Little Andaman and the Nicobar Island. So it's very tough to do the sampling also, but we tried our best to do the sampling from some parts of these islandic groups. So the geological evolution of this island started from the late Cretaceous. That is mostly we get the Radiolarian charts, and we get mostly from, it's known as the Porlob group. So it is the deep ocean marine species, but this late Cretaceous is the base of the sediments found in this island. After this, the basin deepened, and results of deposition, we have some deposition from the late Cretaceous to Paleocene, where we have the ultra-basic institutions and the rocks of Baratan group. Mainly, they consist of dark shells. So this is the late Cretaceous, the serpentinite, and this is the metamorphic rocks from the igneous. The metamorphic rocks are formed from the igneous rocks, and this is the shells that are the dark shells and the siltstone and limestones of the Paleocene period. So after this, we have sedimentation in the late Paleocene to Eocene, that is around 60 to 40 MA. So the deposition is known as the Mithapari or the Baratan form group, and it's shallow water mainly. We have some volcanolithic sandstone, some conglomerates, and the limestone and tuffs. So after this, the Eocene, we have an unconformity due to the plate motions. The plate was colliding, and the Indian plate. So then the late Eocene to Oligocene around 30 to 20 MA. So we have the deposition of the flysch. This is known as the Port Blair group or the Andaman flysch, and this is of deeper environment, and it is thicker of all the sequences that are present in this island. And this is made up of the gray wake siltstone and the shells. So this is all throughout. So this is exposed, and during the high tide, it is been covered by water, and mostly this is continuous to a next island here. This is known as a Ross Island. Like this is Port Blair. This is the capital of the island, and on the right hand side there is a small island, Ross Island. So this flysch continues to that island under water. So then comes the unconformity, as there is, after this sedimentation of the flysch, we have a unconformity, and there was a major tectonic event when the island came up above the water, and we have the Miocene to Pliocene, the archipelago series, and we have most of the biogenic sediments there in this archipelago series. So these are the archipelago series, which is found in the right hand side of the main Andaman Islands. So most of us know that what are microfossils. So it's just less than 2 mm, and we study under the light microscope or the electron microscope. So I will be telling you about the inorganic world microfossils mainly today, about the black box microfossils that I was telling. So that's mostly the calcareous nanofossils, because we can see under the microscope in the transmitted light in the Krosnikov. So it's a black background, and the white calcareous and the calcite crystals blow on the black background. So I recognize it as a black box microfossils. And whereas the radiolarians and diatoms are made, and the silicoflagellates are made up of the silica, and it is clear white in color. So it's a white box microfossils. So, and the forams also more or less white or yellowish white in color. So all this comes under the white box microfossils. So we'll be using those microfossils for my study to understand the past environment and the past climate of this island. So these are the normal methodology. So I hope I can skip this one to. So this is mainly for the in general any paper you can see that. So I will be using both two types of biotic proxies. One is the planktonic and one is the benthic. So the first one is planktonic. I will talk about diatoms, radiolarians, nanoplanktons, and forams. So I will use all these as a age diagnostic microfossils. And for the benthic also, I will be using diatoms, calcareous algae, and the forams. So these are some of the microfossil. This is the diatom. This is the calcareous algae. This is the benthic foram. This is a planktonic foram. This is the calcareous nanofossil. This is the radiolaria. So the major questions of my work is how the planktonic microfossils can be used to relatively date these neogene sediments present in this Andaman and Nicobar basin, and how the diversity of each microfossil can be utilized to understand the paleo environment or the ecology of that region. Was the Indian summer monsoon intensified during the Neogene? As India is dependent on agriculture, so monsoon is a very important climatic event in this part of the subcontinent. So we thought to understand how old is the Indian summer monsoon, and was that any proxy showing us that intensification during the time of that Miocene and Pliocene. And is there any role of the Indonesian through flow in the distribution of the biota in this region? And was there an influx of the tertigenous sediments in this part of the Northern Indian Ocean? Because this Andaman Sea is also termed as the Northern Indian Ocean. So if we talk about the lithology actually, so the Neogene sediments are unconfirmably lying over the Paleogene port layer group, and these Neogene sediments are mainly dominated by mudstone. It may be calcareous or silty mudstone, the chalk, and the limestone. So it is distributed mostly north to south trend. And on the basis of the lithological attributes, it may be divided into two regions. The first one is the northern, and the next one is the southern. The northern region is having creamish yellow chalk with different volcanic ash layers, but in the southern region, we have a calcareous mudstone and limestone, which are devoid of any volcanic ashes. So this is the lithologic distribution, and here you can see the different formations of this basin, and this is the regional stages, which has been correlated with the global stages. So these are the type area and the type sections, and we have tied up with the GPTS age. So these are the different, this is the age, and these are the different chronostratigraphic stages. And you can see that initially in the early to middle Miocene, we have a dominance of the chalk here that is dominated by the calcareous nanofossils and forams. And later on, we have a dominance of the limestone in the middle Miocene, in the Cerebellian period mostly. And then it is again dominated by the mudstone in the late Miocene and early Pliocene. And next to that, in the late Pliocene, early Pleistocene, we have a dominance of limestone again. And in the recent and subrecent, we have the beach sands and the coral rags. So the objectives of the study is to analyze the microfossil content, to understand the relationship between the multiple microfossils, and to understand the precise age, because most of the sediments here are not dated perfectly. So we try to do precise age with multiple microfossils to understand the Neogene paleoclimate and reconstruct the paleogeography, paleo-environment, and paleobathymetry of this island. So initially, I will start about, I will be talking about different time period from this early to middle Miocene, then late Miocene, and early Pliocene, and Pleistocene boundary. These are the ones that I will talk about. The first one is the latest early to earliest middle Miocene. That's the Bardigalian Cerebellian. So with the help of diatoms and radiolarians, you can see this. This is the island. This is the biggest island of this archipelago series. So these are the different outcrops that we worked on. So you can understand that we have to go through sea to collect the samples. So these are the three outcrops, and it's dominated by the creamish white nanophorum chalk here, and the sandy mudstone. So you can see the pictures of the three outcrops. So when we saw the diatoms, there were two zones. One is the Cestoriscus peplum zone, and the next one is the Costodiscus levi-cianus zone. So this varied in the Bardigalian, Langian, until the Cerevelian. So these are the two diatoms. This is the Cestoriscus peplum, and this is the Denticulopsis simonseni, which are the marker diatoms here. So these are the other diatoms which can help us to understand the zone. So this is the Roselia palliaci, which is a marker of the middle Miocene, the early the late early Miocene to the early middle Miocene, and mostly it is dominated by the warm water species. It is very poor. Not these are only the best preserved diatoms I have shown. Mostly it is very poor preservation due to high temperature and effect of dissolution. And with the help of cluster analysis, we have done according to the ecology. We saw that it has been we can divide the clusters, the zones into different bathymetric. One is the deep water, intermediate, and shallow water. One is shallow water, and another one is again intermediate water and the deep water. So these are the radiolarians of that same sections. So the radiolarians are far better preserved than the diatoms, and with the help of radiolarians also we got the similar age as of diatoms. So and the Shannon diversity index also shows that the diversity of radiolarians was much higher than the diatoms during this period, and which was also dominated by the worm species. So this is a WED index which shows us the this is the ecological index which shows us the temperature of the water actually. So this is a warm water dominant species here in this part, and when we are going and like here you can see on the top the shift towards the higher values here when it is coming towards the base of the outcrop sometimes. So the shift towards higher values, that implies lower export productivity, the productivity is less, and this can be correlated with the help of the two papers that which also tells that beyond the 15 MA the middle Miocene climate optimum was there, and the temperature was about 6 to 9 degree warmer than the present. So the temperature was higher, and we also had a previous paper in 2019 which shows us that the MCO event and was dominant, and we can correlate the microfossils with the MCO event during this time. So the paleobathymetry with the help of two different groups of radiolarians. One is the Nacellaria and Spumularia. Nacellaria are the deep water ones, and Spumularia, the red one, are the shallow water species. So mostly it is dominated by the Nacellaria, the deeper water. So we can tell that the deposition took place slightly in the deep water setting to the marginal sea. So the summary we can do later together, so I don't waste time here. So it's then we go to the nanofossils. It tells us NN4 zone, both the all the outcrops, and we can tell the paleo environment is a warm tropical. Here also it shows that it is all the species mostly dominated by the warm water species, and this can be also correlated to the MCO event, the middle Miocene climate optimum. And then it is dominance with the deeper water, deeper to marginal sea, which is also similar to the diatom and the radiolarians. It's a nutricline condition, and deposition was in the epicontinental marginal sea according to the ecology of the species, of the dominant species. The sedimentation rate was really low during this time in comparison to the other one, which we will see later on. It's around 27 meter per million years actually. Yeah. So in the middle Miocene, we don't have any diatoms or nanofossils because it's mostly the limestone dominated. So we can see this is the only part we can access. These are the aborigines stay here, and we cannot. It's a dense forest in this part. So these are the only part where people stay. So these are some of the sections. This is a limestone quarry where we went, and We collected some samples, did the thin section analysis. These are the calcareous algae and very well preserved cells. You can see individual vegetative cells through which we can identify each algal forms. These are some of the planktic forams in the thin sections, and these are some of the benthic forams. It's very hard to identify till species level, so we try to get through till genus level here. So these are the rhodoliths. Actually, these are the algal mats which were present on the rocks in the limestone there. So we can understand that the energy was less, and this can form, the algal mats can form there. So this is the green algae, the halimids, which are also present, very well preserved halimids, and this is the only presence of the halimids in the Indian subcontinent in these islands actually. So we try to do the microfacies analysis, and mostly it is dominated by 3 to 4 types of microfacies, and dominated with the foram and the coralline algal wexstone, packstone, and the drainstone. So we did some geochemical analysis, and it shows that the delta 13C was around minus 20.6 to minus 6, and the average value was around 13, and where the delta 18O is around 1, and so the average value is around 1 parts per mil, and the bulk TOC shows us it around average is 4%, and the delta 13C TOC range from the, so we have done 2 types of analysis. One is from the embedded forams in the limestone, and also from the bulk sediments, to see that if there is any major difference and there is any diagenesis or not. So the nearly constant delta 18O near to 1, it shows that which is near to 0 more or less, which shows that it's exclusively marine in origin and is not affected by any of the diagenesis. The lower delta 13C, you can see a delta 13C around starting from minus 10 to mostly dominated around minus 14 to minus 20, which shows that there is an extra supply of the lighter carbon, and this may be a localized influence in that area, which is produced by the microbial anaerobic methane oxidation. So there is a methane seepage in this area, and this one is not published yet. We are working on the manuscript. The delta 13C values of the calcite sediments reveals that most likely the organisms thrive in the proximity of the hydrocarbon seepages. And when we tried, the isotopic analysis also provides indication that the calcified forams actually be a shallower part of the ocean, and the TOC was nearly high, around more averagely 4%. So it can show that there is a possibility of oil and gas in this region, which is already been demarcated by the ONGC, the oil and gas company in India. So the summary I will talk to you later. So in the late Miocene, we have the so we are talking about Tortonian, because Messinian we have not got in the outcrop, but we have got in a core recently, and it is recently published in a journal by Roy et al. So the diatoms and radiolarians, you can see are very well preserved diatoms in this section. This is made up of the mudstone, and it's very fragile. We could not get the samples from top, because when we were hammering down there, it was falling from top. So we could reach this part around 2 or 3 meters of the section. So these are the well preserved diatoms here. So this one is also about to be published. Work is in progress. So we have around 23 genera and 61 species here, 2 zones. So mostly dominated by the planktonic forms, and the bathymetry is deeper and relatively shallower in the upper part, because it is dominated by the benthic organisms, benthic diatoms. So this is also dominated by the warm water. So, and in the next section, the cave point section, this is demarcated by the the particular radiolarians, Didiomocitris penultima, and Stichocoris delmontensis. And these are the nanofossils which is of NN11A, and the radiolarian zone is RN8 to RN9. So the deposition took place mostly in the deeper waters because planktonic diatoms were more dominant, and with the alteration of the shallow water forms. So this is a radiolarian, particular type of radiolarian, which we observed for the first time in the Indian Ocean. This is known as Acrobotris desolania. So this is as of RN8, and this was previously found in the Pacific Ocean only. So we can tell by this presence that during this time the ITF was, the Indonesian through flow was open, and this has came from the oceanic circulation from this Western Pacific to the Indian Ocean. So these are the diatoms from, this is a very important species, the Thalassonium nischioides, which is the upwelling species. So you can see that we found during the 8 million years, the Thalassonium nischioides with Paralia sulcata and Cosmodus marginatus, all this, this is the upwelling indicator, and high primary productivity is a bloom of Thalassonium nischioides. So we can tell that there is a high productivity due to the upwelling, and this caused due to the Indian summer monsoon was very active during that time, which was also there are some other part which has been in the Arabian Sea also. It has been marked that during these 8 million years the Indian monsoon was very active. So with the help of nanofossils, we have the section is demarcated as NN11A. You can see here, these are the forms. So the sedimentation rate, the last one was 27 millimeter per million year, and here you can see the 200 meter million per million year. So it is drastically changed here due to the monsoon. So when the monsoon, the sediments was dragging from the terrestrial part, it was coming to the seaward side, and it was a low abundance of the discoisters. These are the discoisters here, and the high abundance of the small reticulofenestrates, high abundance. So which shows that there is a coastal upwelling also, so which is similar to the diatoms, and it eventually suggests that there is a nutrient-rich condition there, and high productivity, which may be due to the Indian summer monsoon. Indian summer monsoon is intensified during this time. So during the early Pliocene, we move on to the Zanclean. So here is the Car Nicobar. So this is the only island that we did during the from the Nicobar Island, because other islands are not accessible, and by you have to take several permissions, and reaching this island was also very hard. You have to take a prior permission. As Indian also, we then need to take a prior permission for walking there, because the normal tourists are not allowed. So this is the, you can see that during low tide only we can go down here, but in the high tide the water comes here. It's really hard to do the sampling. So this is the sampling part. So these are the two outcrops where we did, and on the best of the presence of Didymocytris avita, this one, and the Stichocoris peregrina, the Zanclean age was demarcated, and with the help of calcareous nanofossils, we have the Ceratolithus armatus and the Ceratolithus cristatus. These are the two forms which tells us about the age of the sediments here. That is, it is of early Pliocene. So the diatoms or the silicified organisms here are not very well preserved. Most of them are dissolution occurred. So these are some of the best pictures which I tried to make here for you all. So these are the sponge spicules which were much preserved there. These are some of the silicoflagellates which also shows us the environment. These are the calcareous nanofossils which are comparatively better preserved than the siliceous ones. So there is hopefully there is a biogenic silica crash during, and there is a shift in the silica in this part of the ocean. So this is the Ceratolithus armatus, and this is the cristatus here. So you can see when we did the siliceous microfossils abundance, we can see the radiolarians was much dominant, which shows that the diatoms were much fragile and it got dissolution occurred. So we could not find any of the we find less diatoms in this section. So this is a Biogenes silica which was published by a separate author by Cawthon et al. So we saw that they also demarcated that after the late Miocene there was a silica change from the Indian Ocean towards the Pacific and then towards the Southern Ocean. So there is a change. You can see during the 8 million years, 8 to 7 million years, the total silica percentage was much higher than that of the Pliocene. So these are the different planktonic diatoms, and these are the deep water forms and mostly dominated by the shallow water forms here. So this section is really important in this region because you can see most of the studies does not have this Zanclean and here. So only this section can accommodate the gap between the studies of different ODP and IODP cores from this region. So with the help of the calcareous nanofossils, owing to the increase of the discoesters, so in late Miocene, we have less discoesters, but here the discoesters increase, so which shows that it's the global sea level was increasing and during the Zanclean, but the sedimentation was again decreased to 80 meter per million years, which was before in late Miocene was around 200 meter per million years. So there was an intermittent and less monsoon active during this time than the late Miocene. So these are some of the pictures of the discoesters, and these are some of the cuculithophores. You can see the cuculids attached. And the last time I will be focusing on is the Plioplasticine boundary, where we have the calcareous algae and the forams. So this is the Neil Island, a separate island where we have the natural bridge formation. I will show you the picture later on. Hopefully I have here. So these are the very much eroded limestones where it was intercalated by the mudstones, but the mud has been weathered out. So, and it is dominated by the foraminiferal algal grainstone facies actually. Then the age is N21 zone to N22 zone of the foram. So these are some of the aberrated calcareous algae. We cannot see much better cell here, so identification was really hard. And some of the forams were there. So these are some of the ACM we try to do. So the bathymetry with the help of the species of the calcareous algae, the dominance of the Lithothamnium and the Lithophyllum signifies that the paleobathymetry was around intertidal to the 40 meter. So the dominance of the benthic forams also shows that it is the same water depth. There were some agglutinated forams, gastropods, corals, and echinoids presence. So it indicates that it's not much farther into the deeper water. So it is near to the shore. You can see the presence of some planktic forams, which also are not autochthonous. It is being carried from the deeper waters through the wave, and it is allochthonous deposition actually, or the para-autochthonous, you can tell. So this is the section of the Plioplasticine boundary. It's just somewhere here. So we tried to do the planktic forams. So these were much well preserved. So this limestone is far from the water. So the water comes maybe the highest high tide just here. So the water can't reach here. So we have a soft limestone here. So we try to do the chemical preparation and got the. So we have two species. One is the N. pachyderma, another is the N. deuteratri. So the N. pachyderma is cold water. So you can see that when we see the ratio, the deuteratri and the pachyderma, so we have a interchange of the cold and warm water during this Plioplasticine boundary. So this is the natural bridge that I was talking to you about. So it's the several sea notches are also present. So we can walk only during the low tide. So we need to go came from this side and came here for the collection. So one sampling was from here and one from here. So we have different subpilute tubes also. So which we tried to do some analysis that it's diagenetic or not, but we saw that it's not diagenetic. And some stabilizotropic studies also shows that there was a high negative delta 13C similar to the middle myosin. So we can tell that there is a cold seep activity, a methane seep during also near to this area. So, and this area has also been demarcated as a frontier basin of India, actually, gas and hydrocarbon. So they have not used it up, but later on, maybe in the future, they will use the oil and gas prospective from this region. So just to conclude, I will take 2 minutes more. In the latest early to middle Miocene, we have the age demarcated through different microfossils. The siliceous microfossil shows that it is, so the age is around Langian to the Cerevellian, that is the late early to the middle Miocene. The reduced abundance here, we have less abundance of the siliceous microfossils, and this may be due to the dissolution, which is a characteristic, maybe a characteristic of the low latitude warm water during that time, and the poor preservation of the diatom valves may be due to the high temperature and effects of dissolution during the time of the MCO event, actually. So with the nanofossils also, we saw that it is a nutricline condition and warm tropical condition. So the plantic benthic ratio of the diatoms shows us that deposition took place in the deeper water environment. In the middle Miocene, we have the larger benthic forams indicate that it's upper to lower photic epipelagic zone, because we also have the different types of algae present there. So these algae can only sustain in sunlight. So there is a well-littered area and shallowest part of the upper photic zone mainly. So we have rhodoliths. I show you the algal mats. So that are the rhodoliths. So they only sustain in moderate energy settings. So it is moderate energy there, and the delta 13 values shows us we are near to the hydrocarbon seepage. Late Miocene shows us that based on the multiple microfossils, revealed us the age is Tortonian, and the planktic diatoms, the planktonic bloom was much dominant, and there was upwelling during this period, and which shows that the high productivity, and there was intense monsoon during this late Miocene in this part of the world. So next we come to the early Pliocene, which is much dominated by the warm shallow water taxa. So, and the radiolarians are the main contributors to the biogenic opal. Diatoms were much less, but there was dissolution to the siliceous microfossils, and this maybe this can tell us that the water temperature changes, which was induced by the monsoons in the late Miocene. So it has been developed periodical gyres or local upwelling, which changes the abundance of these microfossils. So there is a shift in the opal accumulation from the equatorial region to the Antarctic region, which can cause this widespread biogenic silica crash during this time period at 6 MA, and also maybe to the closure of the Indonesian through flow. So the Pleistocene boundary, we can see the early Pleistocene and was indicate high energy condition with turbulent. So there was some upwelling and taxa. So this upwelling and the presence of the cold water can tell us that the Pleistocene glaciation also started during around 2.7 million years. So from here we can also tell that the Pleistocene glaciation may be started after this. So thanks a lot for I acknowledge my institute, my previous institute, BSIP India, my fellowships which I availed during there, and also some of the part of the work on the nanofossils was done at UCL at the lab of Professor Paul Baum and Dr. Jeremy Young, and some of the forums work was done in collaboration with Professor Bijay Wed. So, and at last, I need to thank Andaman and Nicobar administration to help me to get permissions to work out in this island, where normal people cannot go in some of the islands where we got permission and can visit there to collect the samples. Thank you very much. Thanks a lot again to Paleopucks for inviting me. Thank you. And I'm happy to take any questions.
Thank you very much, Arindam, for this great presentation. Very interesting to get some information from, like you said, this location that's usually not very accessible. We do have a few questions, so as usual, I'm going to post them in the chat and read them out loud as well. So we already have a few. So the first question that we have is from K. Sender, and he asks, why is the Andaman fleece very thick? Is it due to merely high sedimentation rates or events concerning the formation of the Himalayas and changes in the Bengal Sea fan?
So the Andaman fleece is mostly much thicker because the sedimentation was really coming from the Himalayas time. So Himalayas was forming and it was eroding at the same time. So the sedimentation was much higher during that time, and also the thickness, because it's much harder than the because later on the sediments was much fragile. The archipelago series, you can see the mudstone which was much eroding, so we don't have as much of the sedimentation during the archipelago preserve, but mostly the fish was dominant due to that the quality of the sedimentation, because it's mostly the Radularian cherts or some of the shells and fish. Actually, it's much harder to sustain the weathering also.
Thank you. I think that explains the question very well. I have another one here, and it asks, does the presence of the planktonic diatoms in the Messinian, seen for the first time in the Indian Ocean and previously observed only in the Pacific, indicate strong south oscillations or the ENSO system and stronger oceanic currents in the region? I hope I read that correctly.
Yeah, I think we didn't got any Messinian diatoms yet, because in my presentation I'm not shown, but the Messinian one I just told because it was we got some because I am not part of that paper from my same lab, but they got from the course actually, but we don't have any Messinian deposits in the outcrops in Andamans. We didn't encounter any Messinian diatoms yet. So I think we have till late Miocene in the Tortonian, so we have the sedimentation.
Okay, thank you. We have a couple of more questions. Again, if you have a question, please send them to the questions host. I think this one was already a bit addressed, but do you think that there could be any sampling or preservation biases between the different types of microfossils? For example, siliceous versus calcareous?
Mostly all the where I got the siliceous, I got the calcareous also. So it's very different actually when you get both the things in the same type of sediments. But in the limestones, we don't have, because it's much harder. The limestone side is much harder, so that it's not fragile limestone. In the hard limestone, we don't have any, because diatoms are only, siliceous microfossils are only preserved in soft sediments. It's not preserved in hard sediments. So it's not a bias. It's a type of sedimentation which preserve that type of microfossils actually. But in the soft limestones, we have encountered the only forams, but we didn't got any calcareous nanofossils. We tried, but we didn't encounter any of the calcareous nanofossils. I don't know, it has been dissolution occurred or it is not being preserved.
Okay, thank you. Then we have another question that again goes back a bit to the beginning of the presentation, I think. So with these two different sedimentation types, does the bifurcation between the northern and southern neogene sediments, for example, i.e. the extensive deposition of volcanic material in the northern regions, but not the southern regions, correspond to the paleocurrents and wind patterns, pushing and concentrating the volcanic ashes in the north?
Yeah. So in the southern side, I mean, it is the Nicobar Island. It is much down, where from the, I think it's a part of the Toba ash, volcanic ash, as recently in the IODP 354, they also encountered this type of volcanic ash in the northern part. So hopefully it is a part of the wind pattern which deposited in the northern part, not in the southern part of the island.
Okay, thank you again. We have a bit of a broad question. So can these findings or conclusions be applied in modern times to better understand modern climate, climate change, and its biotic effects?
Can you please repeat the question one?
It's also in the chat, but I will read again. Can these
Okay.
findings or conclusions be applied in modern times to better understand modern climate, climate change, and its biotic effects?
Yes, because most of the this study was done in the Neogene time, the Miocene and Pliocene, which can be correlated with the, because the islands are in the same position still, and most of the ecology of this of these microfossils that I worked on is the present ecology only. We try to correlate. So definitely we can use these findings to the modern times, as our climate, our temperature is more or less similar. Like in the middle Pliocene, you can see a 3 to 4 degree centigrade more than the present climate. So hopefully we can correlate this study with the modern climate and the climate change, and the different biotic effects also, what we are experiencing currently. But we need to do some modeling studies for that. Directly we cannot correlate to the modern one.
Okay, thank you. We still have more questions, so I hope you're ready. So this one is, how important are terrestrial runoffs from the islands in supporting the microbiota and determining the taxonomic distribution of the planktonic microbiota?
Okay. Because you can see mostly when there is a deep water sedimentation, it's oligotrophic. But in this case, you are finding lots of nutricline condition and lots of nutrient in the water. So definitely it came from the nearby. So the eastern side of the island. So if this one is the island chain, so west side is the Indian side, and the eastern side is the from the Thailand. So there is a major river which is flowing in the west side of Thailand, that is Irrawaddy. So the terrestrial runoff from that river mainly contributed to the archipelago series, with supporting the microbiota during this Miocene and Pliocene time, and determining the taxonomic distribution of the okay, so mostly it's a planktonic because it is a deep ocean setting. So we are getting a planktonic microbiota. Only the nutrient is coming from there, but it is not the organisms are coming flown from there. So it's a autochthonous deposit that we are getting. When we are getting planktonic microbiota, that means it's an autochthonous deposit of that island. It is not being flown from the terrestrial part of the Irrawaddy Delta or from the shallower part of the Irrawaddy Delta region to the Andamans. So we are pretty confirmed that these planktonic forms are present there. So it is only the nutrient is increased, and these planktonic forms are formed there in these islands.
Okay, thank you for your answer. We have one more question, which reads, great talk. Could you correlate the periods of weaker and stronger summer monsoons with insolation changes, for example, like Milankovitch forcing?
Yeah, I understand, but I have not done any with the summer monsoon with the insolation changes yet, because need to do more work for that. Because for this I need to work on the isotopes, but still our isotope work is very poorly done. So we are working, because I'm not expert of the forams, and some of my colleagues are working. So if we have more isotope data, then only we can do this insolation changes correlation with the stronger and weaker monsoons. I'm very sorry, but hopefully later on in coming years you will be seeing the results from our lab.
Okay, thank you. Always good to have something to look forward to. I think those were the questions, but we're also at the hour already. So I want to thank you again for your presentation. Again, I want to confirm, would you like to skip tea time or do you still want to stay after this?
I can stay for 15 minutes like that. Is that okay? If someone want to
Yeah.
interact, it's definitely okay. I'm happy because it's only 12, so I can stay till 15 minutes more.
Okay, that's fine. Thank you.
Yeah,
Then
thank you.
I am going to share a bit more announcements. So hang on. So I hope you can all see my screen. Yeah, so again, everyone who has been listening here, thank you for tuning in. Again, we would like you to fill out the weekly feedback form, which has been posted in the chat. Then I would like to point your attention to the next week's seminar, which will be by Bram Vaas from the University of Milano Bicocca in Italy, and which will be about mapping the past towards quantitative paleogeographic reconstructions for all paleosciences. So I hope to see you next week as well. And then before we go into tea time, I have one more announcement. So we are actually looking for colleagues or people who would like to join this great committee. So we are looking for 1 or 2 new members right now, with specifically people with an interest or experience in speaker scheduling or grant writing and fundraising. But if you don't have experience with these specific topics, we will still really encourage you to apply if you're interested. Because we're not necessarily looking for experience, more for motivation. So the link for that will also appear in the chat. The deadline is May 3rd next week. So we hope to hear from you. And with that, I would like to go into tea time. If you are familiar with tea time, normally we have a countdown, but I actually removed this from the slide because I thought that we wouldn't be having tea time. So if you want, you can take a 2-minute break where I turn off your screen if you want to grab a drink and then come back. Thanks, everyone.